All right, are there any questions? So last time we were talking about programmed cell death, all right, or cell senescence. So let's just wrap up that discussion and then we'll move on to the next chapter, all right? So a couple of things to note regarding cell senescence. Reactive oxygen species especially hydrogen peroxide, can serve as internal signals to promote senescence. Another promoter of senescence could be high concentrations of sugar. Right? So high concentrations of sugars can also serve to signal leaf senescence. And this is especially true if nitrogen content is low. So especially if nitrogen levels are low. Now let's talk about, like we've talked about various plant hormones, right? Which ones among those promote senescence and which ones basically inhibit senescence, right? So senescence promoting regulators so among the plant hormones, ethylene, so we discussed a total of 9 plant hormones, right? So ethylene, abscisic acid, then jasmonic acid, then brassinosteroids, and salicylic acid. And then the senescence repressing regulators, that means the ones that prevent senescence from happening. So what do you think they'd be? What are the things that we have left out here? Auxin, cytokinin, gibberellin, right? All those guys. So cytokinins, auxins, and gibberellins. Now, abscission is the process of some organs falling off, like leaves falling off from plants, okay? Fruits falling off and all that. That's referred to as abscission, right? So abscission is the shedding of leaves, then fruits, then flowers, or other plant organs. And this kind of abscission, it is brought about by programmed cell death, right? Because the cells that are attaching, let's say, the leaf to the rest of the plant, they will die, okay? There's going to be programmed cell death, or they will senesce, and then the leaf falls off, right? Very, very neatly, right? So this is brought about by programmed cell death. In the abscission zone. That means in the zone of cells where the abscission is going to take place. Right? It's got some special layers in them because again, as you may have guessed, all of this has to do with gene expression. Right? The cells must be having the right gene expression in order for them to undergo programmed cell death and then for the leaves and other organs to fall off. And again, this has to do with the activity of hormones. So if you have high levels of auxin, then the tissue is going to be insensitive to ethylene. Ethylene is a promoter of cell senescence or ripening or even programmed cell death, right? But if you have auxin active over there, then it's not sensitive to ethylene, right? So high levels of auxins implies leaf tissue is ethylene insensitive. However, if the level of auxin drops, right? If auxin level drops, then ethylene sensitivity will increase and the leaf will fall off, right? So ethylene sensitivity increases and leaves fall off. Now, as I mentioned yesterday, right, you have programmed cell death at the cellular level, right? You have senescence at the organ level, you have senescence at the plant level, right? But at the molecular cell level, all these things involve the same kind of mechanisms, right? So at the cellular level, programmed cell death, then organ senescence, and whole plant senescence use same or similar genetic pathways for cell autolysis. So what is autolysis? Lysis means to split, right? Auto means just by itself. So it's basically cell senescence, right? So they use the same mechanisms, right? Now if you're looking at plants, some of them are annuals, right? Some of them are biennials, right? So some of them will produce fruits, flowers and seeds only once, then they will die, okay? Some of them are there all the time, okay? So they will go through many seasons of flowering and producing seeds, right? So they differ. So the ones which basically flower in 2 years, okay, the lifespan is a couple of years, they are called biennials, all right? So the first year they'll probably spend in growing, right, vegetative growth, and the second year in reproductive growth, right? So annuals and biennials reproduce only once, right? For the biennial, the reproduction will be in the second year, right? So they reproduce only once before senescing. That means the whole plant dies. While perennials can reproduce multiple times before senescing. Now if you're looking at what are called monocarpic plants, right? That means the plants that reproduce only once during their lifetime, right? As soon as the reproduction is over, right, it's time for the plant to die, to exit the stage, right? So for those plants, whenever the nutrients start moving away from the leaves to the seeds to the reproductive structures, right, That's a signal for the induction of senescence, right, and plant death. So nutrient or hormonal redistribution from vegetative structures to reproductive sinks may trigger whole plant senescence in monocarpic plants. And these are plants that reproduce only once during their lifetime. So reproduce only once. All right, any questions? And this makes sense because once the reproduction is over, the plant has no business hanging around, right? Because the next generation has already been created. So any questions on this? Otherwise, we'll now move on to the next chapter, right? And I think all these chapters that I've been lately covering, you can see that there's a lot of signal processing involved, right? And there are lots and lots of problems that, masters and PhD students can work on. You want to model gene regulatory networks. There is some information that is available in the literature about the pathways, but it's not complete, right? So you can put that information together, produce your network, make predictions, see if they work out, and things like that, right? So the next chapter, again, even that will also involve all these signaling pathways, right? That's chapter 23 is about biotic interactions. So that means interaction of plants with the living organisms, right? So chapter 23 is biotic interactions, right? Now, see, when you have a plant, right, it's going to be attacked by pests and pathogens and so on, right? But so the plant will have to mount its own defense mechanisms, right, against plants, against pests and pathogens. But the pests and pathogens also will try to survive, right? So they are going to mount some counter defense mechanism against the plant's defense mechanisms. Okay, so it's an ongoing war, right? It's like a guerrilla warfare. No side is completely eliminated, right? But it continues because you're seeing plants all around you and you're also seeing the pathogens. Okay, so there is a co-evolutionary race. So co-evolutionary race between plants and their pests. And these are mainly herbivores. That means organisms that eat up plants, right? And pathogens. Now, not all interaction between plants and other organisms is bad, right? We have seen the example of nitrogen-fixing bacteria, okay, where there is a symbiotic relationship between the bacterium and the plant, right? The bacterium is providing the plant with what? Nitrogen, yeah. While the plant is providing the bacteria with sugars, all right? So beneficial interactions are there, right? So beneficial interactions between plants and microorganisms and examples are nitrogen fixing bacteria. And we have talked about how these nitrogen fixing bacteria, they release what are called NOT factors, and these interact with the plant and they lead to the formation of nitrogen fixing nodules, right, on the plant root. So formation of nitrogen fixing nodules, right? Similarly, you have other bacteria that are called rhizobacteria, right? The rhizosphere is the space surrounding the root, right? So and the bacteria living there, those are rhizobacteria. So rhizobacteria can release metabolites that assist plant growth by, number one, increasing nutrient availability, because the bacteria can make changes to the local environment, right? So some of the nutrients become now available to the plant roots, right? So by increasing nutrient availability, and number two, by producing some antibiotics, right, that will kill harmful organisms, right, in the vicinity of the root. So producing antibiotics for protection against plant pathogens. So these are the beneficial interactions between plants and other living organisms. But you can also have harmful interactions, right? So harmful interactions between plants and pathogens and herbivores. And the first line of defense for the plant, okay, against these harmful interactions is mechanical barriers, right? So many of these plants are equipped with thorns, then spines, prickles, trichomes, and rhaphids, right? And the book has details about each of these things, okay? But these are, like if you have a thorn, if a herbivore goes and tries to eat up that plant, it'll get thorns stuck all over in its throat, okay? So there are problems, right? So it'll run into problems. So this is like a deterrent, right? So rhaphids are mechanical barriers that provide a first line of defense against herbivores. Then in addition to that, right, you could have some secondary metabolites, right? Primary metabolites are things that the plant needs just to, survive, to carry out the basic functions, like all the enzymes that you need for glycolysis, TCA cycle, and so on, right? Those are primary metabolites, right? Those are enzymes that you need. But secondary metabolites are things that the plant may not need, okay? But if it's attacked by some other herbivore, then it's going to make use of those, right? So secondary metabolites they are stored in compartments. Why? Because if the plant is producing those toxic secondary metabolites, it could be harming itself, right? So the things are separated. The different components are separated. When the herbivore comes and eats that plant, right, things get mixed. Okay, there's damage, and then the toxic stuff is produced, right? So secondary metabolites stored in compartments. These are released upon damage. And there are several examples of that. I just give you a couple. For example, if you look at the hemlock plant, okay, that's used to produce opium, right? It'll have neurotoxins in it, right? So hemlock produces neurotoxins, right? Then, so if a herbivore goes and eats up hemlock, they'll probably have spasms that could even cause death, all right? Then another plant called foxglove produces digitoxin, right? Which interferes with cardiac function, because it is going to go and mess up the sodium potassium pump, right? The sodium potassium ATPase pump, because this pump is responsible for maintaining the concentration of sodium and potassium in cells. Okay, so they have to be kept at certain levels, right? So continuously there's work going on, right? So now if some herbivore goes and eats that foxglove plant, right, and if it eats it in the right dose, okay, it is going to mess up cardiac function, because the heart cells, they need that, for them for even to generate the heartbeat, okay, that sodium potassium ATPase has to function, right? So they interfere with cardiac function, leading to increased myocardial contraction. And these molecules, all right, that are produced by these plants, they also have medicinal value. Because if somebody is suffering from congestive heart failure, then you can give them medicine made from this, okay? That's going to alter the activity of the sodium potassium ATPase, right? So leading to increased myocardial contraction. Now some of these metabolites are also stored in vacuoles, right? So some secondary metabolites are stored in vacuoles as water soluble sugar conjugates. Right, so they are stored in a form, right? They are there, but they're not active all the time, right? If some herbivore comes and eats up part of the plant, there is damage. In response to that, these molecules are released, okay? So that is going to have a negative effect on that invading organism or invading animal, right? Now the responses that I talked about so far, right, using all these secondary metabolites, right, and even the thorn spines and all that, these are what we would call constitutive responses, because they are already there, right? These molecules have already been produced. It's just that there has to be some damage or insult or something like that for the activity to take place. So these are all constitutive responses. So these are constitutive. Right? These are all constitutive responses. Now what's the disadvantage with these constitutive responses? Is the plant losing anything by, uh, providing for these constitutive responses? Yes, it is, because the energy is required, right? Energy resources are required to produce these all the time, right? It's like in anticipation of war, you're in a war mode, in a state of heightened alert all the time, right? So that's not good, right? So there should be some responses that are also inducible, right? If there is an attack, if there is a pathogen attack, then the response is going to be induced, right? So the next thing I want to talk about is inducible defense responses to insect herbivores. So inducible defense responses to insect herbivores. That means plant-eating insects, right? So here, the defense is produced only in response to the stimulus, okay? So here produce defensive compounds only in response to mechanical damage or in response to the insect saliva. The insect saliva might have certain compounds, right, that could trigger the response, right? So mechanical damage or insect saliva. And these are called elicitors, right? Or the stimulus, right? And the advantage is that this is going to save resources. So you're not producing these defensive compounds all the time, right? Now, how does that happen? Well, insect damage, and again, there will be signaling, right? So insect damage leads to rapid increase in jasmonic acid. So there's a jasmonic acid synthesis pathway, right? That has to be activated, right? So the level of jasmonic acid is going to go up, right? So insect damage leads to rapid increase in jasmonic acid, which induces transcription of plant defense response genes. Right? And obviously, if there's transcription taking place, so there are genetic regulatory networks involved, right? There's scope for doing microarray studies, RNA-seq, and so on, right? So I'm just mentioning that. So scope for genome-wide studies. Example, microarrays. Then next generation RNA-seq, etc. So the same kind of techniques that we talked about earlier, they can be used here also, right? To get the overall picture and hopefully do better than you would do just looking at one gene or one protein at a time. Now, herbivore damage can induce system-wide response also. Not just the local response at that point, but all over the plant it can produce responses. So how does that happen? So let's see. So herbivore damage can induce system-wide response For example, there is a peptide, a polypeptide called cysteamine. Cysteamine, it's a polypeptide. Is released into the apoplast, right? What is the apoplast? Yeah, it's basically the cell walls, the space between the cell walls and all that. If it's released into the apoplast, that means it can travel all over, right? Go to other cells, right? So released into apoplast, so that can induce jasmonic acid synthesis at distant sites. So those sites, the cells at those sites will know that, hey, there is a herbivore that is out on the loose, all right? So start preparing your defense chemicals, right? Then plants also transmit electrical signals. So plants also transmit electrical signals to stimulate responses at distant sites. In addition, they release volatile compounds to attract the enemies of those herbivores, right? Like if it's a caterpillar that is eating up the plant, right? So the plant is going to release something that is going to attract other birds and all that like to prey on that caterpillar, right? So release volatile compounds to attract uh, herbivore enemies. And also alert neighboring plants and alert neighboring plants that there is a herbivore nearby, right, that is active. So they have their, like their code, we have the code maroon, so it's the same thing, right? Plants have that too. All right, any questions so far? So we talked about plant defense against herbivores, okay? Now let me talk about plant defense against pathogens, right? So microorganisms that invade plants, right? So what are the defenses that are employed by plants against them? So plant defense against pathogens, right? So if you have a plant cell, right, it's going to have a cell membrane, right? It's going to have a cell membrane, right? And then see the way the cell is going to recognize that there is a pathogen is because maybe the dead cell walls, okay, of the pathogen are there, right? Or some molecules like that, right, that are recognized. So on the cell membrane, you have what are called pattern recognition receptors, right? So there is a pattern recognition receptor or PRR. So pattern recognition receptor or PRR, right? And this pattern recognition receptor is going to recognize what is called a microbe associated molecular pattern. That means something that is left over from, let's say, dead pathogens, right? So it's going to recognize this molecule that is called, or this signature, which is called a microbe associated molecular pattern. So this is MAMP, right? So the PRR is going to recognize the MAMP, right? And it knows that if that MAMP comes and binds the PRR, then it needs to get to work, right, to produce all the defensive compounds, right? So there is going to be some signaling inside. Receptor-like kinases, all right, receptor-like proteins will be involved, right? So we have done this kind of thing for animals before, right? When we talked about cancer, growth factors, and all that stuff. Same kind of thing happens here, right? So there is signaling, and that involves receptor-like kinases, then receptor-like proteins, right? And the net result will be the production of these anti-pathogen molecules, right? So this is going to produce defense response molecules. So produce defense response molecules, right? And this so it's like in response to that microbe associated molecular pattern that is detected, right? The plant is producing those defense response molecules. So this is like a kind of immunity, right, that the plant is acquiring, and this is referred to as MAMP, microbe associated molecular pattern triggered immunity. Right? That's one aspect, right? But see, if every pathogen that comes in, okay, is subjected to this MAMP triggered immunity, right, then all the pathogens will have disappeared, but we still see them around, right? So what does the pathogen do? Well, I mean, the pathogen is also smarter, right? It'll figure out a way because it knows that if it tries it going in through those pattern recognition receptors, all right, is going to get killed. So instead of doing that, you bypass those transmembrane proteins and, using a syringe, just inject stuff directly into the cytoplasm, right? Bypass this pathway. So typically the pathogen, the response that it will evolve, right, is bypass that pattern recognition receptor and directly inject into the cytoplasm, right? So directly inject what are called effectors, right, into the cytoplasm. So pathogens directly inject effectors into cytoplasm, right? Now if the plant didn't figure out a way how to deal with this, right, then the plants would have disappeared, right? So the plant produces some molecule, right, that is going to recognize that effector, right, and then produce some other, proteins and genes, right, to take care of this invading organism, right? So pathogens directly inject effectors into cytoplasm. The plant is going to detect that, and that leads to the production of capital R, which is for resistance proteins. So there is transcription involved, right? So resistance proteins. And so this type of immunity, right, against the invading pathogen is referred to as effector triggered immunity. And obviously this is an ongoing battle between the plant and the pathogen, okay? You think you're smarter, you come up with something, the other invading organism is going to produce something else, right? And the procedure continues, that's how they are able to evolve, right? Otherwise, one or the other would have been eliminated. All right, any questions? So this is one main mechanism, right? And then another anti-pathogen defense is what is called the hypersensitive response. So when the pathogen infects, let's say a leaf, all right, the cells in the neighborhood of the infection site, all of them decide to undergo programmed cell death. They're signaling. So if they die, right, then the pathogen has nothing to feed off, right? So the reactive oxygen species level will go up, the pathogen will die. And so it's like those cells have sacrificed themselves for the overall good of the entire organism, right? So another anti-pathogen response another anti-pathogen defense is what is called the hypersensitive response. And in this hypersensitive response, what happens is that cells surrounding the infected site rapidly undergo programmed cell death, so PCD, thereby preventing the infection from spreading. All right, any questions? So this concludes our discussion of the chapter on biotic interaction. These chapters are pretty easy to read at the end. Okay, I'm just giving you the summary from the end of the chapter. Having read the chapter, I can go through the summary, fill in some details. But if you're interested, you can go and read that. Okay, because that's not difficult. Because it's not like what should I say? It's more about signaling and all that. It's not about hardcore biology, like where you have vegetative growth, organogen. It's like it's not the kind of biology that we as engineers would like to spend too much time reading, right? But these chapters are different, right? This biotic interaction and even the next chapter on abiotic interaction, because it is about signaling. There are some networks also that are given there. It's just that I didn't have the time to, put those up here. Okay, any questions? So now we move on to the last chapter of this book, right, which is chapter 24. and this is about abiotic stress or abiotic interactions. Biotic means that is brought about by living organisms, like if you have a pathogen that is attacking or something like that. Abiotic could be heat, drought, salinity, and so on. Okay. Now, stress, plants are not the only ones that encounter stress. All right. Animals also have detailed stress response mechanisms. All right. So, for example, if oxygen is limited, right? So you could have a low oxygen level that would they would refer to it as hypoxia, right? If you have hypoxia in an animal, right? The same thing is true even in plants, right? You should switch from the TCA cycle to glycolysis, right? Because you have to save oxygen. You cannot use oxygen. So there are different stress responses like that, you know. So plants and animals respond to stress using dedicated stress response pathways. So stress causes the transcription of what are called stress response genes, which result in alleviation of the stress. For example, right, if you have oxidative stress, okay, what is oxidative stress going to produce? I don't know whether I mentioned it or not. Sorry. Suppose you have oxidative stress, all right, that is going to produce one. What? No, no, that's not exactly a stress thing. The ethanol is because of the glycolysis, okay? But if you have like too much of oxygen, right, or let's say hydrogen peroxide, what is it? Hydrogen peroxide is what is referred to as reactive oxygen species, okay? So oxidative stress will result in the production of reactive oxygen species. So oxidative stress results in ROS or reactive oxygen species production, which activates the production of ROS scavengers. All right, there are molecules that will basically kind of take care of these ROS molecules, right, before they can cause any harm. So oxidative stress results in ROS production, which activates ROS scavengers. For example, superoxide dismutase, and there are others too, right, that will go ahead and basically kind of defang these ROS molecules before they can do any harm, right? Now, what is stress? So formally you can define stress. So stress can be generally defined as an environmental condition that prevents the plant, and again this we are defining in the context of plant biology, condition that prevents the plant from achieving its full genetic potential. Under otherwise ideal conditions. For example, in the context of plants, drought could be a stress. It's a water stress. You have too little water, right? So example, drought then you have excess salt in the soil, right? So salinity, etc. Right? And I will talk to you in a few minutes about how these things can mess up different parts of the plant function. You know? Now, plant responses to abiotic stress, right? And in this chapter, we are talking only about abiotic stress because we already covered biotic stress in the previous chapter, attack by herbivores, pathogens, and so on, right? So plant responses to abiotic stress, they involve trade-offs between vegetative and reproductive growth. Yeah, obviously, like if the plant is stressed, okay, and let's say you don't have enough nutrients and all that, right? You don't go and reproduce, right? If you're barely able to survive or take care of yourself, you don't go and bring in more offspring, right? So a plant does that same kind of thing also. You know, the question is, should there be vegetative growth or should there be reproductive growth? Should you produce flowers, seeds, and all that, or should you just produce more leaves so that you can capture enough sunlight, right? And do enough photosynthesis to barely feed yourself, right? So now, in the context of stress response, right, of plants, there are two terms that are used. One is adaptation, the other one is acclimation. Adaptation means it changes over many generations, right, to repeated stress, right, and so there will be genetic changes in there, right. Acclimation means it's something that, you know, maybe of a single plant, right, that has been exposed to, let's say, heat or cold or whatever. So it's done some tuning, but usually, or in many cases, the process is reversible, right. It's not across generations. So let's make that distinction. So adaptation is genetic changes in the entire population that have been fixed by natural selection over many generations. And acclimation is basically individual plants. It's not the entire population, so it's individual plants responding to periodic changes in environment so this can alter either the physiology of the plant or form, right? That is morphology. Like if you have a plant that is like growing in a very hot area with a lot of sunlight, right? So it might produce leaves. It might end up producing leaves with smaller area, surface area, right? Just in order to survive. So that would be an example of an acclimation response. Right? So individual plants responding to periodic changes in environment. So they will alter either the physiology of the plant or form, that is morphology, that's the change in the leaf that I was talking about. And these physiological changes require no genetic modifications. And they are reversible in many cases. Now let's look at a few examples of abiotic stress responses, right, and what the plant can do to basically counteract the effect of these stresses, right. So examples of abiotic stresses, right. And you will see that all this stuff actually makes sense, right? The first thing that I want to note is that most of the stresses, all right, they might result in different responses, but at the end of the day, the stress is going to lead to the production of reactive oxygen species, right? That's almost like a universal feature of stress, of different stressors. So most stresses result in the accumulation of ROS, right? Now let's look at few stresses that and this ROS can also damage DNA, right? Remember, we need correct copies of the DNA, right? This ROS can go and mess up some of those bases, right? It can produce adducts in them, and even in animals it can cause cancer too, right? These oxidative damage can cause cancer, right? So one example of a plant stress is cell dehydration, right? So what is going to happen if you have cell dehydration, right? Not enough water. So there's going to be decreased turgor pressure, right? So decreased turgor pressure. Then there's less water, right, in the cell. That means there'll be ion toxicity. The concentration of ions will build up. So, so decreased turgor pressure, increased ion toxicity, and if you have less water, right? See, remember that when you have photosynthesis, right, for every molecule of carbon dioxide you absorb, you have to send out 400 molecules of water. If you have less water, that means you can do less photosynthesis, right? So, and reduce photosynthesis. Then the next kind of stress that we are going to look at, or we're going to talk about, and again, in the book there is a section devoted to each of them. So if you want more details, you can take a look over there, right? But you're getting the big picture and things will be making sense, right? The next stress that I want to mention is salinity, right? That means too much of too much of salt in the soil, right? And the plant is absorbing too much of salt, right? So what is going to happen? Now, in the presence of salts, okay, some of the proteins will denature, right? They'll lose their three-dimensional conformation. I talked about that before, right? Right at the beginning of the semester, that changes in pH, salt concentration, they can cause protein denaturation. That's not good. You need a functional protein, right? So it can cause protein denaturation. Then it can mess up your cell membranes. So membrane destabilization. And when that happens, right, you're going to have reduced above ground growth, right? Because the plant is under stress, it's not going to grow that fast. So reduced above ground growth, and also if you have reduced above ground growth means you'll have lesser number of leaves and so on, right? So the photosynthesis will be less, yeah. So reduced photosynthesis. So none of these effects are good, right? All the things that are happening are things that we don't want, right? So this is salinity. Then the third one is light stress, right? So if the plant is getting too much of light, right, sunlight, more than it can handle, it can mess up the photosynthetic apparatus, right, produce reactant oxygen species and so on, right? So here you have plants receiving more sunlight than they can handle, and this will lead to ROS production. Then temperature stress. The temperature is too high or too low. Again, temperature stress is going to mess up protein stability, right? Like if you look at an egg, right? If you boil the egg, what happens to that protein outside? It all becomes white. Okay, it's different, right? I mean, so temperature stress is going to mess up protein stability. It'll mess up, and protein stability messed up means enzymatic reactions, all right? Let's say high temperature Rubisco is there. Okay, you're supposed to use it for doing photosynthesis. It got messed up because of high temperature, right? So it's not going to be able to function as an enzyme, right? So enzymatic reactions will also get messed up. Then membrane fluidity. Remember, the membrane of the cell is made up of what? Of what? Lipids, right? So if you have too many saturated fatty acids in there, they will freeze at a higher temperature, right? So you have a problem because this has to be a permeable membrane. Things can have to go inside and be able to come out, right? Through all those channels, pores, and things like that, right? So there is an issue there. So temperature stress can cause loss of membrane fluidity, and it can also mess up the structures of DNA and RNA. Then another kind of stress, all right, if you have flooded soil, all right, the soil is flooded with water, the roots are not going to get oxygen, right? That's the problem. Okay, so flooded soil, aerobic stress in the root, right? And you can refer to it as anoxia. That means it's not getting enough oxygen, right? If it gets less oxygen, that's hypoxia, right? Anoxia or hypoxia, but that can kill the plant, right? Potentially. Then you can have freezing stress, right? If you have freezing temperatures, right? Below freezing temperature, right? Now remember, inside the plant you have the xylem and the phloem, right? So you need liquid flow inside, right? Liquid and all these solutes and all that have to flow. If you have freezing temperature, if your pipes get frozen, guess what? You're out of luck, right? So freezing stress is a problem, right? So it will lead to cell dehydration because the water is not going to be communicated to all the portions of the plant. So cell dehydration. Xylem can be blocked. Even phloem, but xylem can be blocked, right? Then another kind of stress that you can have is from absorption of heavy metals, right? Like arsenic and all that from the soil, right? So that's a problem too. Because that heavy metal is going to go and substitute, let's say, for iron or something like that, right? And with iron in that molecule, let's say in chlorophyll, all right, you have iron. You need iron over there. So instead of that, if arsenic or something else gets in, okay, it's not going to have exactly the right shape for the function. So the plant is not going to be able to carry out the functions that it normally needs, right? So heavy metals. So they will displace other key metals and disrupt key reactions. Then if you have lack of mineral nutrients in the soil, that's also another kind of stress. But in that case, the plant is not going to be able to grow, right? Because if the essential nutrients are not there in the soil. So lack of mineral nutrients, this is going to suppress growth. and also reproduction, right? So if you don't have enough nutrients, right, the plant shouldn't be reproducing, right? It can barely feed itself, right? Then another kind of stress is produced by ozone and ultraviolet light. Again, this results in the production of reactive oxygen species and also in induction of programmed cell death. So ROS production and programmed cell death. Because the ultraviolet light is going to lead to DNA damage. We talked about that before. Even in plants, the same thing will happen, right? Now, the last thing that we want to note, right, in this listing of stresses is that combinations of stresses. So if you have like temperature, right, then you have too much of light, flooding, right? The combination of stresses, right, is going to produce responses that are different from those individual stresses just combined together, right? So this is a nonlinear phenomenon. It's not like you superimpose the stresses and you'll get something linear, right? And those different stress pathways also interact with each other. So combinations of stresses produce effects different from those of individual stresses. So this is multivariate, right? The interaction between the different stresses is multivariate. It is multivariate. And the biologists refer to this as crosstalk between stress signaling pathways. And even these pathways, they are not completely known, right? I mean, some of the portions may be wrong. In case of plants, I think it's a little bit better than in case of cancer or something like that, human disease like cancer. Now, another thing to note is that plants can gain cross protection, right, when sequentially expressed, sequentially exposed, not expressed, exposed to different abiotic stresses. Right? And this is because of crosstalk. Like you might have, treated the plant for heat or something else, okay, and then you find that, you're getting a better response for some other stress that you did not expose it to, right? That is also observed, right? And that's because there is crosstalk between these different pathways, right? Now, how do plants sense stress, right? So stress sensing is carried out in many different ways, right? So stress sensing, it can involve physical, biophysical. Like if you're talking about light stress, right, that is going to be sensed by photoreceptors, right? So physical, biophysical, then metabolic. Maybe not enough oxygen is available for doing the TCA cycle, right? Metabolic, biochemical, and epigenetic mechanisms. And again, epigenetic was methylation, acetylation, and so on, right? Not changes in the genome, right? But basically going and turning something on or off, right? So epigenetic mechanisms to detect stress and activate corresponding pathway. Now, stress actually initiates in most of these stress response pathways, right? Not just in plants, but I'm more familiar with the ones in animals, right? Stress initiates transcription of stress response elements, right? And I can very briefly, actually I cannot probably talk about it because we're running out of time. So that's okay. Let me just stick to the script, right? Um, Now, as I said before, many stress response pathways share signaling intermediates. And to see that, I would refer you to some figures in the book. Where you'll see there are common signaling points between many of these pathways. So see figures 24.9 through 24.12, right? So they've given the detailed diagrams for some of these signaling pathways, stress signaling pathways. All right, so now let me talk about some of the mechanisms that provide abiotic stress protection, right? So the stress is detected, then you have some signaling going on, right? What are the mechanisms that the plant uses to protect against different kinds of stresses, right? So let's talk about them. Mechanisms providing abiotic stress protection. The first one, it can lower root water potential, right? It can start, increasing the solid concentration over there, maybe release some starch from the vacuoles, right? So then it becomes sucrose, right? That's going to reduce the water potential, and it can still draw water in the drying soil. So lower root water potential uh, to continue absorbing water in drying soil. Then when there is flooding, root flooding, right, it can develop special parenchyma tissue, right, which basically allows it to send oxygen to the flooded organs, right, or submerged organs. So develop parenchyma tissue to supply oxygen to submerged. And again, all of this, each of these responses is going to involve a lot of signaling, right? Then if you're faced with oxidative stress, you have reactive oxygen species that can be detoxified through scavenging mechanisms, right? So you have to send in something that is going to eat up reactive oxygen species. So through scavenging mechanisms. Then if you're having protein that is getting denatured, right, you can make use of chaperone proteins. The plant can produce chaperone proteins. So chaperone proteins prevent sensitive protein denaturation. Then if you're having a change in temperature because of which the membrane is going to freeze, right, the lipids, the plants can actually alter the composition of that membrane, right? If you have more unsaturated fatty acids, then the membrane will stay fluid, right? So the plant can alter composition of membrane lipids. to retain fluidity. Then if you're talking about uptake of toxic heavy metals, all right, the plant has got two options. Number one, it can selectively not take them up, all right? Or if it takes them up, it has to have internal mechanisms, all right, to get rid of the harmful effects, all right? So exclusion and internal tolerance mechanisms for toxic ions. Then if you have a situation where ice crystals are being produced inside the xylem, right, then the plant needs to produce some antifreeze molecules, right? So generate antifreeze proteins to prevent ice formation. Then if the plant is losing too much water, okay, it's really very dry, there's a drought situation, right? So the drought causes the production of abscisic acid, right? And abscisic acid is going to cause stomatal closure, right? So stomatal closure by ABA, that is abscisic acid, induced efflux. Efflux, that means exit, right? Efflux of, not influx, efflux of potassium and anions from guard cells, right? So here the stimulus is drought, right? It is not light, right? In the presence of blue light, we know how water flows into those guard cells and then the stomata open. Here the water will flow out because if potassium ions and anions they move out, right? Then the water potential will become high. Water goes out, right? So and the stomatal pore closes. Then the plants can also alter leaf morphology and root to shoot biomass ratio, right? Which I mentioned earlier. If there's too much of light, the plant can alter the shape of the leaf, right? So that the area is smaller, right? Shoot to root biomass ratio, if the nutrients are limited, right? Then the roots need to grow more, right? In search of those nutrients, water and so on, right? Now, suppose the plant has been subjected to stress, okay, and it has, activated the stress response pathways, right? If you suddenly remove the stress, the plant is going to die, right? It needs a gradual reduction of that stress, right, so that it can slowly adapt to the new conditions, right? So reversal of stress response pathways must occur gradually so that you don't start producing too much of ROS, right? So no ROS production. And an important goal of agricultural research, right, is to develop crops with enhanced stress tolerance, right? So if you want to grow plants with less water, right, it should be drought resistant, right? You want to grow plants, let's say even for biotic stresses that are resistant to pathogen, right? You might want to engineer some of the genes in such a way that the plant can do a better job when the pathogens invade, right? So develop crops with enhanced stress tolerance. So this is where ideas from engineering, all right, are going to merge with ideas from like plant biology, right? Because right now what people are doing, it's like one gene, one protein, right? They went and bred some seeds, they produced some seeds, they think it's drought resistant. It works in many of the cases, all right? But if you want to do a better job, you want to optimize, right? Then you're going to have to develop more detailed models and work with those, right? Which is the gene that you should knock out to maximize your chances of getting better yield, right? Or produce a plant that can survive with less water, right? Now, here I should mention that I gave you one paper, right? It is there on eCampus about modeling the MAP kinase pathway, right? In the context of plant pathogen interaction. So right now you're in a position to read that paper. I gave you all the biology, right? And that's one of the first papers that we worked on. And working with people in other disciplines is not easy because they do not move at the same rate, right? The time scales are different, right? So they were looking at combination of different stresses. Like the question is, how are the genes going to behave, right? Different genes. Okay, how is gene expression going to be altered if, let's say, you subject a plant to drought stress, okay, and at the same time have it attacked by some pests, okay, like some herbivores, all right? So which are the genes that are expressed, right? So we worked on that, but it was taking them a long time, and it's not like we are in sync. So I told my student, right, that we need to do the theoretical work without waiting for the data to come, right? Because you guys have to graduate, right? I mean, like my student needed to graduate. She cannot wait 5 years, or because the other person messes up, right? So we are in a position to do that. Like, I mean, use data from the internet and all that, and do theoretical work when it's absolutely necessary, right? But the ultimate goal, like for PhD students, would be like take a problem of interest, okay, motivated by agricultural considerations, all right? Do the modeling, right? And then propose something, and hopefully, by the time you finish your PhD in 4 or 5 years, we can get it tested out. Because the facilities are here on the A&M campus. Like if you drive along 47, right? Sorry, not 47. If you drive along 60 towards the Brazos River, A&M has lots of land over there where these kind of control experiments can be carried out, right? So that's one advantage that we have here. And of course, I mean, the cancer research also is there, okay, but that's separate from what we are teaching in this class, right? Now, regarding the stress response, right, yeah, I can spend like a couple of minutes and just tell you about that. Like in the case of animals, I know that these different stress responses, all right, the way it occurs is that there is there are some sequences in the DNA, right, inside the nucleus. Those are called stress response elements, right? So normally there is something that is working as a brake on that, so that those genes are not transcribed, right? Now, if the plant if the plant or if the cell is stressed, right, then it releases a molecule. So there is a combination of something that is called a sensor, right, and a transducer, right? So something is sensing the stress. So normally the sensor and the transducer, they are held tightly together outside the nucleus, right? So in response to the stress, right, these molecules let go of each other, right? And one of them, right, will go into the nucleus, all right, and it is basically going to start the transcription of that stress response element, all right? And that produces the response, and there is a negative feedback kind of effect because as the response is produced, all right, you don't need to mount the stress response. So there has to be a negative feedback effect that will cause the response to get reduced as the effect of the stress is dissipated, right? And for plants, it's not explained in that much detail in this book, but my guess is that it's no different, right? It's similar, okay? So this is what I wanted to tell you about. I had thought that I could even discuss that paper that I mentioned about plant pathogen interaction, but we are really running out of time, so we have to stop here, And any questions? So let me